Hi, I'm Alok Finn from CV Path Institute and University of Maryland. I would like to thank CardioNovum for inviting me to give this talk. I'm going to talk about DCB coating materials and technology, highlighting key differences in distal embolization. I think an interesting and important topic in the world of drug-coated balloons. Here are my disclosures. I think I'm going to begin with a more basic slide. What are the elements of an effective drug-coated balloon formulation? I think it's important to understand this. The DCB must deliver large quantities of drug within seconds. Obviously, it's different than a stent. It in the course of 60 to 120 seconds, must deliver the drug effectively to the target site. That drug must distribute within the media within the first few hours to days. And the therapeutic drug levels must be maintained for more than 1 month, ideally 3 months overall. I think these are important issues. And then, of course, one major concern with drug-coated balloons is effective drug delivery to the target tissue while avoiding non-target effects. And by that I mean minimizing emboli to downstream tissue beds where we're really not supposed to have drug. We have current CE mark and FDA approved drug-coated balloon devices for coronary artery are listed, and you can see here among the devices, of course, almost all of them are paclitaxel devices, and what differentiates them is the excipient as well as the drug dose. You can see the doses tend to vary by drug type, and of course the type of paclitaxel employed in the balloon also is important, highlighting, of course, the Restore and Restore Plus by CardioNovum, which has more of an amorphous form of paclitaxel. We'll discuss that in a minute. And, of course, the newer sirolimus-coated balloons, which were just highlighted at TCT, including the Solution and Magic Touch. These two have U.S. IDE studies underway and are probably the more advanced sirolimus-coated balloons. So this slide is a more basic slide, but meant to reinforce the idea that a DCB is not a DES. DES, of course, are, I would say, somewhat easier to make these days because, of course, we have the metallic scaffold, we have the polymer, and then we have the drug. And the scaffold itself is the basis for the polymer-drug combination, which delivers drug over long periods of time. And, of course, we have that as a base of drug delivery. Whereas for DCBs, we've got to there's nothing there, of course, when we deliver the drug. So the drug has to be transferred to the wall very quickly, as I mentioned before. And there has to be some mechanism for retention of the drug within the arterial wall that keeps it there and makes it sustained over time. And of course, the drug has to be excellent diffusion. And for stents, overall distal emboli are not a concern because the stent and the drug really the stent holds the drug in place and it really only treats the target tissue. Whereas for DCB, it really depends on the coating type in terms of embolic debris. So, of course, we have two drugs, sirolimus and paclitaxel. They work differently. They're both effective anti-restenotics. Paclitaxel works by inhibiting mitosis and is considered more of a cytotoxic drug for the most part at doses we use, actually killing cells, but being very effective in neointimal formation in terms of prevention. And then, of course, there's sirolimus, which we consider a cytostatic drug, paralyzing cells in G1 to S phase, and really not causing as much cell death as more cellular paralysis. Now, paclitaxel, it's important to understand paclitaxel processing if you're going to understand paclitaxel-coated balloons. There are really two forms of paclitaxel. It's a complex organic drug having multiple solid structures as well as crystal and amorphous terms. The crystalline form of paclitaxel is really the form that's used in most drug-coated balloons today, and the amorphous form is less employed. Now, the crystalline form has advantages in the sense that, of course, it forms almost like a drug depot. So the drug retention over time is better with the crystalline form. Its biological effectiveness is stronger because you've got more drug releasing for longer periods of time. But there's more chance for particles release because it's a crystal, remember, and it's got more chance for vascular toxicity than the amorphous forms. And this is shown here on this slide from this paper where we show pharmacokinetic levels of drug in tissue. And you can see here in the solid line, the crystalline form of paclitaxel simply has longer drug indwelling in the wall versus the amorphous form where you can see the dotted line. The drug levels quickly start to drop by 24 hours, 7 days, and 28 days, almost undetectable, whereas they're very easily detectable with the crystalline form. You can see the differentiation from a paclitaxel-coated stent, PES, there shown. Now, retention, how does paclitaxel work? Well, you can see in this animal model where we've deployed a paclitaxel-coated balloon, the crystals themselves are really deployed on the surface of the artery wall where they stick, and they're really going to cause a fibrin mesh almost process, and they start to diffuse into the artery wall, and that's how paclitaxel has its effects on preventing intimal formation. And to say that these are not successful would be wrong. If you look at different drug-coated balloons that are paclitaxel, and these are peripheral balloons, Ranger, Impact, Lutonix, you can see all of them have fairly good paclitaxel levels out to at least 45 days in terms of pharmacokinetic profile in animal models. And you can see that really depends on the actual dose of drug used. The Admiral DCB Impact has the highest dose of drug, and you can see its kinetic profile is a little longer drug retention than something like Ranger. And then Lutonix has the least amount of drug retention because it has the lowest drug dose. Now, coating integrity of these paclitaxel-coated balloons is variable because, of course, each of them are different, and each of them have different constituents. You can see that the crystalline coating on paclitaxel actually is easily shed when you blow up the balloon at the bench top, depending on the different type of balloon. And of course, you have to think about this in the context of what happens in the arterial wall when you blow it up. Of course, you're going to have some drug transfer to the wall, but of course you're also going to have some release of drug into downstream tissue beds. And it's like a crystal. When crystals are cracked, they shatter, and they cause shards to form, and those shards can go downstream. Now, this slide is an important slide that shows you that paclitaxel adherence to the balloon, this is showing two different coatings, but the real message I'm trying to show here is that there's a lot of drug split off during dry inflation. It's not as if the drug is holding on to the balloon well in these coatings. A lot of it's lost during passage, and a lot of it is lost during the inflation. And the remnant on the balloon, you can see there's still some paclitaxel left, but it's said that only 10 to 15% of the loaded dose of paclitaxel probably makes it to the wall. So it's important to understand the inefficiency of drug-coated balloons. Now, drug transfer efficiency, as I mentioned before, is shown here for the BD Lutonix balloon and the Medtronic Impact, and you can see that about 83% of the drug loaded is lost during the procedure, 12% or 14% retained on the balloon, and in this particular example, only 5 to 3% is transferred to the vessel wall. It's said that it's better with sirolimus-coated balloons. Here I'm showing you solution data. That remains to be proven, but in this particular slide, yes, there was better transfer to the vessel wall with the sirolimus solution product. Now, this is the particulate, what the particulates look like. You can see we did scanning electron microscopy. You can see what the balloon surface looks like with Restore. It's actually fairly smooth there, probably because of the amorphous coating. Sequin Please has more of a rough coating. It has more crystalline paclitaxel, and you can see that the actual particulates themselves range in size of most drug-coated balloons, paclitaxel that is, from 100 to 400 microns. And we say anything greater than 100 microns can actually lodge in downstream arterioles, causing tissue ischemia. So this is a concern with the paclitaxel-coated balloons in terms of distal emboli, and this is what the actual particulates actually look like under high-resolution scanning electron microscopy. Now, the animal models are very good for trying to understand this process because they really allow us to deploy the balloon, for instance, in the SFA, and look downstream at downstream skeletal muscle and what's called coronary band, which is a vascular structure that connects the shin to the hoof of the animal, and assay those areas, those tissue areas, for not only drug quantitation, but also presence of emboli by histology. So it's very useful to look at how much drug actually went into downstream tissue beds where we didn't actually want it. And this was a schematic of a study we did a number of years ago, randomly assigning animals SFA to Impact, Lutonix, or POBA, and looking at not only the artery wall, but the downstream effects on non-target tissues. And you can see histologic parameters for evaluation of DCB. We really look at things like proteoglycan replacements, muscle cell loss shown on the upper right, and fibrin deposition, all as indicators that the drug is actually working in the vessel wall. Animal models cannot really tell efficacy, but they can tell drug delivery by using indirect signs of drug effect, and these are the ones that we primarily rely on for paclitaxel-coated balloons. Now you can see it's a low injury model, as I mentioned. There are really no differences in terms of the way Lutonix and impact perform on the arterial wall, and you can't really use this as a measure of efficacy, but you can use this as a measure of safety, and you can see that all the balloons are fairly safe in terms of their effects on the arterial wall. There is proteoglycan replacement seen there, but there's no other effects in terms of inflammation. Downstream incidence of distal embolization is shown here in this original study we performed looking at Lutonix versus impact, and you can see that even a single balloon had a significant evidence of distal embolization in the percent of sections. You can see, of course, impact has more drug dose than Lutonix, and was seen in about 25% of sections. Emboli were seen by histology, and then when you use overlapping balloons, 3x dosing, of course, that tends to go up even further. And you can even see these emboli present at 90 days in the downstream skeletal muscle. Now, this correlates also with downstream drug. If you look at the skeletal muscle and coronary band after treatment with Lutonix or impact, you can see it's much greater for the impact than Lutonix, and those drug levels are still detectable to some extent at 9 days. So it's important to understand that the emboli as well as the drug detection in the downstream skeletal muscle is still can be found when you do drug-coated balloon procedures with these balloons. And lest you think this is just relevant to those two balloons, you can see this with other crystalline paclitaxel balloons. Here I'm showing you data from another leading DCB. This is Prevail, showing you downstream evidence of distal emboli. The way we detect them, of course, is by looking at the downstream. This is myocardium, I think I'm showing you here, which is showing you areas of inflammation with surrounding fibrin and in small arterioles, and perhaps you can oftentimes see under polarized light either balloon material or even fragments of crystalline paclitaxel, which shows you evidence of the embolic debris. Now, some people ask, well, does it make a difference if you use a stent? Can you have a stent, and does a stent cause emboli? Well, you can see that in this particular animal study we did and published, you can see the only way we saw emboli was to use a paclitaxel-coated balloon. When we used a POBA plus Zilver PTX, which is a paclitaxel-coated stent, we saw no emboli. Whereas when we use DCBs, regardless of the use of BMS or just the DCB plus Zilver PTX, we saw evidence of downstream emboli. So it's the balloons themselves that cause these downstream embolic changes. Now, vessel safety and embolic potential, this was another study I conducted. This time I wanted to compare the results in terms of distal emboli with the solution, Magic Touch sirolimus-coated balloons, and agent, which is as you know, a crystalline paclitaxel-coated balloon, which is made by Boston Scientific. And in this methodology, of course, the porcine coronary arteries were implanted with the balloon, and we went back 28 days later and scored not only the arterial wall, which I'm showing you how the methodology is done in terms of histologic scoring, but also the downstream downstream myocardium. And you can see we can sample certain areas of the myocardium downstream of the LAD, the circumflex, and the RCA to figure out how much emboli occur. When I say emboli, we actually look for two things: evidence of infarction or microscopic scarring or necrosis as the most severe finding, so muscle actually dying or scarring, or evidence of embolic or foreign material in downstream arterial small vessels. And you can see in this analysis that the agent balloon was the only balloon that caused evidence of myocyte necrosis and scarring versus the POBA, and both sirolimus-coated balloons didn't have that effect. You can see that emboli were also seen with all drug-coated balloons, as probably would be expected, but it was greatest with the paclitaxel agent-coated balloon and least with the POBA, as you might expect. Even POBA, though, does have some evidence of emboli, but no evidence of myocyte scarring and necrosis. Now, you say, and some people say, well, what's the importance of all these findings? What does it mean? I don't see this clinically. What does it matter? Well, I think it does matter in the sense that emboli are important. And one area where emboli may be the most important because of flow dynamics is the lower extremities. This was, as you all know, DCB for BTK lesions has actually never been approved. No DCB is approved for below-the-knee interventions. And in this particular study, paclitaxel-coated balloons from the impact deep were actually associated with greater rates of amputation. So there is a lot of concern about what these emboli mean, especially for areas of the body like below-the-knee interventions. And so I think it's important to not dismiss the findings I just highlighted to you. And then we know in coronaries, for instance, when you use paclitaxel-coated balloons, these are evidence of transient slow flow phenomena. I think if you talk to operators, regardless of the location, coronary or peripheral, slow flow is seen in some interventions, up to 10%, they say, in some interventions. And that's obviously a concern. And the microcirculation coronary flow reserve is also impaired to some extent by using these paclitaxel-coated balloons. Probably not surprising given the experimental data I just showed you. Now, we do know slow flow is associated with lower rates of wound healing and lower major amputation-free rates. Here you see if you have slow flow after the procedure, you have less good wound healing and you have lower amputation-free rates. So I think flow dynamics after DCB angioplasty are extremely important. So sirolimus, why do we not have a sirolimus-coated balloon available, or why didn't they come more quickly? Obviously, there's difficulty with sirolimus because the reliability is compromised by inefficient drug delivery from the balloon surface, and a dedicated vehicle is required. Tissue absorption is limited following acute drug transfer. Sirolimus degradation occurs quickly, and you probably need some sort of encapsulation, and it deactivates quickly in tissue with a short half-life. So you need some sort of carrier, and then you need to transfer that carrier to the wall and have that dedeployed over time. You obviously want to minimize distal embolization. So sirolimus-eluting balloon requirements, you want to increase drug uptake, you want to extend drug retention in the wall. Sirolimus must be continually delivered over time. So you need some sort of time release mechanism. And you also need some sort of instant glue that binds that sirolimus to the wall. And you also want to limit embolization drug wash-off. Now, this was a study conducted in the porcine model looking at crystalline sirolimus. What happens if you just use the crystalline form of sirolimus? Well, it's somewhat effective in terms of inhibition of stenosis. You can see here the p-value was not significant for diameter and area of stenosis. So just using the crystalline form is probably not very effective. You need some way to have a drug depot and to bind that drug to the vessel wall. Now, there's some sirolimus-coated balloons here. I'm just going to introduce two. The Concept Medical solution really involves phospholipid drug carrier, which encapsulates sirolimus. These are submicron particles, about 0.4 microns, and they have sirolimus in them, and then those are transferred to the vessel wall in this phospholipid carrier. Now, solution is a little more complex. It has a proprietary microreservoir technology. It has PLGA polymer, which is admixed with sirolimus. And then what happens is there's a phospholipid or coating that helps to bind that. It's an emulsion almost that's made with the microreservoirs and the cat. And the cat helps to bind that to the balloon surface, and it optimizes its transfer to the vessel wall. So this is a two-step process. And in terms of clinical data, we certainly have some data from sirolimus-coated balloons. This Transform 1 clinical trial was done where we were compared Magic Touch to the Sequent Police paclitaxel-coated balloon, and we looked at the outcomes, and you can see that the sirolimus-coated balloons didn't quite perform as well as paclitaxel in terms of late lumen loss at 6 months, but they still perform fairly well, but not quite as good as what was performed with the paclitaxel. Now, solution has a little bit longer drug release, and it performed better in first-in-man studies. You can see the late loss was excellent. In the SFA, it was 0.19. In the coronary, it was 0.16. So I think overall, solution may have a little bit better angiographic results. Now, microvascular resistance in humans. This was a paper by Patrick Sroy's group who looked at whether microvascular resistance is changed after sirolimus-coated balloons or paclitaxel-coated balloons. And you can see here, he was actually able to show, similar to what we showed in animals, that sirolimus-coated balloons had no change in microvascular resistance. It was angiographic microvascular resistance. And you can see, whereas the paclitaxel-coated balloon, the angiographic microvascular resistance went up after the intervention significantly, suggesting, again, the presence of emboli. Now, the Solution de Novo trial was actually presented at TCT. This was a very interesting, provocative trial where basically patients with de novo disease, excluding STEMIs, were randomized 1-to-1 to a strategy of a DES per normal way you would implant a DES versus a DEB arm where they were basically enrolled and then they underwent optimal vessel prep and underwent the DEB if the vessel prep was adequate, and that's defined in the paper. And then they were looked at 1-year TLF rates, and we looked at whether there was non-inferior result compared to the stent, and actually turns out the result was non-inferior. That was presented at TCT just a few days ago. So in conclusion, I would tell you there's no DCB class effect. Differences in dose, drug, structure, excipient all influence the effects. Crystalline paclitaxel DCBs all have potential, embolic potential, but the consequence remains uncertain. But there is data emerging to show physiologic effects, and in animal models, certainly these emboli and effects are detectable. Size and character of the particulates differ from DCB to DCB. Sirolimus DCBs are emerging, but they're not as well characterized as the paclitaxel DCBs, and I think the technology will continue to evolve as time goes on.